Hello, my name is Dr. Sarwat B Rizvi and I would like to present an overview of the paper titled A Novel POSS-Coated Quantum Dot for Biomedical Application. This project was supervised by Mr. Mohammed Kashgar and Professor Alexander Sefelian. Just to look at what quantum dots are fluorescent semiconductor nanocrystals of the size range of 1 to 10 nanometers with unique photophysical properties that give them tremendous advantages over the traditional fluorophores and organic dyes. Quantum dots can be used for a wide range of biomedical applications, particularly cancer diagnosis and therapy. For example, they can be used for cancer localization, detection of micrometastasis, image-guided surgery like sentinel lymph node biopsy, and image-guided delivery of chemotherapeutic drugs. However, the main limitation to their biomedical application is their toxicity, as most QDs are based on heavy metal cores of cadmium, tellurium, and selenium. In this paper, we explored the possibility of using a novel polyhedral oligomeric silicious chioxane nanoparticle as a biocompatible surface coating for quantum dots. POSS is a caged silicious chioxane with an inner inorganic framework of silicon and oxygen and an outer organic framework. The overall size of the POSS nanoparticle, inclusive of its organic surrounding, is 1.5 nanometers, which makes POSS the smallest of the silica nanoparticles. We have previously integrated the POSS unit into a polymer that we've synthesized in our lab called the POSS PCU and used it for various tissue engineering applications like vascular grafts, cardiac stents, nerve grafts, as well as more recently as a nasolacrimal duct and a tracheal graft that have successfully been implanted into human subjects. The advantageous properties of POSS in polymer systems include an enhanced resistance to biodegradation and hydrolysis, enhanced biocompatibility, and reduced immunogenicity and enhanced antithrombogenicity. In this paper, we integrated the POSS unit along with mercaptosuccinic acid and D-cysteine onto the surface of cadmium telluride quantum dots, which was synthesized by a one-pot technique. The quantum dots were characterized using transmission electron microscopy to determine core size, fluorescence spectroscopy to determine peak emission wavelength, and a Fourier's transform infrared spectroscopy called FTIR for determining the surface chemistry, as well as to confirm the bonding of the POSS unit to the QD surface. This is an ATR FTIR machine, and the POSS and MSA QDs were placed as a drop on the diamond ATR window, and the spectra were recorded. The diagram shows the appearance of a new peak at 1043 centimeters on the POSS QDs, which is absent on the MSA QDs, indicating that the POSS had actually bonded to the QD surface. Our results demonstrated that POSS QDs had an enhanced colloidal stability and a significantly enhanced photostability on exposure to UV radiation for a period of 2 hours. In vitro toxicity testing was conducted using HepG2 cells for an exposure time of 1 and 24 hours, using both the two different types of QDs. We found that both POSS and MSA QDs were significantly less toxic than ionized salts of cadmium and tellurium, while the toxicity of the two QDs was comparable. The most interesting outcome of this study was a significantly enhanced brightness of the POSS QDs after intracellular uptake in HepG2 cells at both 1 and 24 hours. And this led us to believe that a lower concentration of POSS QDs may be required for in vitro imaging applications, leading to lower toxicity and overall enhanced biocompatibility. Based on the results of this study, we can conclude that a novel POSS coating confers enhanced biocompatibility, enhanced photostability, and enhanced colloidal stability to the core cadmium telluride quantum dot. It also imparts features of amphiphilicity, which may lead to enhanced uptake across lipophilic cell membranes. Future work on this study relates to determining the in vivo biodistribution of POSS QDs, as well as conjugation to specific antibodies for targeted localization of cancer cells both in vitro and in vivo. Thank you.